Hi guys. So today I'm going to be talking a little bit about my research, and then we're going to um, get into a little bit about COVID, um, since those were the requests we had. Let's see, can I pull up the chat and do this? How old am I? Probably too old. If you have a question, please feel free to stop me, and then Stephen
Yeah,
or
okay.
Celeste, can one of you interrupt me if there's something in the chat that I don't see? Uh,
Yes.
okay, cool. All right, so I study neuroscience and I study dopamine neuron subtypes and how they interact with chronic pain and addiction. So Um, yeah, and then Northwestern always has us do this. I have nothing to disclose, and that just means I don't make money from any of my discoveries. Um, okay, so I know that there's a big pandemic going on, and we'll talk about that in a little bit. But before this, you may have heard of something called the opioid epidemic. And this is a map that I think really drives home sort of part of that epidemic, which is painkiller prescriptions. So opioids are painkillers. You might be familiar with fentanyl or morphine are two classic examples. And so this map is just showing how many painkiller prescriptions there are per 100 people. And so in the yellow, you can see that even in the lowest states, um, of which Illinois is one, we're looking at 50 to 70 painkiller prescriptions per 100 people. And then in the sort of most extreme examples, it's up to 143 painkiller prescriptions per 100 people. And this is from 2012 because that's the most recent year that data is from. But all trends indicate that this has probably gotten more extreme. And so this is just something to think about when people talk about the opioid epidemic. And yes, there's heroin use, but a lot of it was really driven by prescription drug use and abuse. And so I think that this really shows just how many of these drugs are being prescribed. Um, so when people talk about the opioid epidemic, something that gets lost often is why on earth are people prescribing so many opioids, or doctors prescribing so many opioids to their patients? And so something that was shocking when I learned it is that chronic pain is the most prevalent and the most common sort of disease state in this country. Um, and as you can see from this graphic, more people live with chronic pain than cancer, heart disease, and diabetes combined, and that is estimated to be up to a third of the country living with some sort of chronic pain disorder. And so one of the reasons that doctors prescribe such massive doses and so many opioids to their patients is that so many of their patients are truly suffering. And unfortunately, unlike cancer, heart disease, and diabetes, there isn't actually 20 years of solid research on this topic. And so when opioids don't work, or um, in 100% of cases when they have high abuse potential, you still use them, often because we don't know of other treatments because it really hasn't been studied. So once it started, people started looking at it. They started doing fMRI brain scans of people. And so that's where maps like this come from. And this map is just all areas that have been associated with chronic pain um, in some way. And then the blue bubbles are the classical reward network. So these are classically implicated in addiction. And I study the ventral tegmental area, which I'm going to call the VTA for the rest of the time, because it was named anatomically before anyone knew what it did. So its name doesn't actually tell you anything about its function. And this is a major area that has long been associated with reward and addiction. and separately was determined to be associated with chronic pain. So that's why me and my lab are interested in it. And okay, here's another map. And so now what you're looking at is that these blue tracks are the two major dopamine nuclei of the brain and where they project to. And the reason this map is kind of important to think about is that not every neuron in the brain makes dopamine. There are actually very few neurons in the brain that can make and release dopamine, and they are predominantly located in these two nuclei. This nucleus is called the substantia nigra and is mostly associated with motor coordination, motor function, and motor decision making. And this area, which is the area that I study, the VTA, as we just discussed, is associated with reward and reward decision making. And as you can see, has very far-reaching axons that go all over the entire brain. Specifically, the track that I'm interested in is this little tiny track right here. And this little star is over the nucleus accumbens, which is a downstream target of the VTA. And this dopamine from the VTA to the nucleus accumbens is the dopamine that they talk about in the press when they talk about reward, and dopamine being a happy molecule or rewarding molecule. That's this dopamine. So that's the dopamine that I study. Okay. So all of those images were in people. So let's just get a little bit coordinated in mice, because all of my work is done in mice. It's quite invasive and it would not be ethical to do in people. And as you can see by this chart, the green dopamine from the VTA to nucleus accumbens is the only dopamine in this circuit. This is a pain circuit. And the other thing to notice is the mu opioid receptor, which is just a receptor that responds to endogenous opioids, but also exogenous opioids like morphine, and that is also located in the VTA. And so when we talk about opioids and morphine specifically having abuse potential or addiction potential, this is one of the mechanistic reasons why we think that is. That these receptors have a function in an area that we know in general is associated with reward and addiction. Okay. All right, Pavlovian and classical conditioning. I don't know if you guys have heard of this before, but this is a learning paradigm. And so, all right, so now you have the background of sort of where we are in the brain, what we're looking at, and why we're looking at it. This is a little bit of a practical brief on what kind of behavior and stuff we investigate in my lab. So classical conditioning is just this idea that you can associate a neutral or otherwise meaningless stimulus with a reward, or actually with something negative, but we're talking about reward today. So this was originally discovered by a guy named Pavlov, or whose last name was Pavlov, and what he noticed is that his dogs, when he gave them food, would drool. And when he gave them a bell or a tone, they had no response. However, if he always gave them the tone before giving them the food, eventually the tone itself would elicit the drooling even before the food was presented. And so this was the original finding that indicated to Pavlov that the dogs at least were learning and they were associating the tone with their reward. And so for a long time, there was a search to find, okay, what in the brain is responsible for that? And like, how do you know? How do you eventually make that connection that this tone is going to mean food? So this is from a seminal paper in 1997 from Wolfram Schultz, and we're going to go through it pretty slowly. But in each panel, there's this lower box that looks like it has random dots. Each row is a single neuron recording, and each blip is a neuron firing. And at the top,
I guess there is just one question, so
yeah,
I'm not sure if you would have mentioned this later on, but Winston wants to know how different the dog's brain would be in comparison to a human's brain.
a dog brain in comparison to a human's brain.
I think that was the question, or like when it comes to this sort of testing, conditioning, would there be more similarities or differences?
Yeah, so that's a great question. Actually, this has now been tested in almost every model animal and even in people with fMRI. And it has great cross species conservation, which means that in dogs, mice, in monkeys, which actually this recording was done in monkeys, and in people, it seems to elicit the same brain responses in terms of the analogous area in a dog versus a person versus a mouse. Something that you might have noticed, for example, in this brain, you can see the cerebellum. This is our brain, right? You can see the cerebellum is here, which is like back of your head there. In a mouse, the cerebellum is in a different spot. It's up here. But it can still be recognized by both, in this case, by striking anatomical similarities, but also we do identify neurons by molecular and other physiologic measurements. So for example, we know this is the VTA in part, or the analogous VTA to what we have in humans, in part because it's the locus that makes dopamine. And so, yep. So so in fact, in people, if you had a dinner bell, you would actually produce saliva earlier in preparation for digesting your food. Okay. All right. So like I said, in this panel, in this lower section, each row is a single neuron and each dot is it firing. And then up top, this is the conglomerate data. And then the conditioned stimulus is given at this time point. And believe it or not, these are all aligned papers. This is the original figure, so forgive me for the formatting. And then this bar and this point down all through here is where the reward is given. So the conditioned stimulus is that tone that we were talking about. And then the reward, in this case, is actually little chocolate pellets, because like I said, these are monkeys. Okay, so this is before any training has happened, and you can see, okay. So who are these neurons? These neurons are the dopamine neurons of the VTA. And before any training, you can see that when a reward is given, when they get the little chocolate pellet, you have this sudden increase in activity in the dopamine neurons. So great. So they're responding to reward, but do they only respond to the reward, or do they help you learn? So the next test is to see, once the monkeys are trained, do they respond? And they in fact do, right? You no longer see this peak after the reward is given. You only see it right after the conditioned stimulus. So that means that these neurons are part of the circuit that's helping you learn. And it actually turns out that they're critical to this circuit, and that this part of the learning is totally dependent on them. Okay, but there's one more little tidbit which is sort of interesting, which is that what happens if you give the tone to a trained monkey, but then you don't give them any chocolate pellets? What happens then? So in that case, what you see is that you still get the response to the conditioned stimulus, which makes sense because the monkey doesn't know it's not going to get pellets. But then once it doesn't get any pellets, you actually see a decrease in activity, which is different from up here. When you get the reward and there's no response in these neurons anymore, it is actually a pretty sizable decrease, and you can see in the single neuron data how obvious that is. And so this is one of the reasons that dopamine is always associated with reward, even though, as you can see, the amount of dopamine is actually telling you information about reward, predicted reward, and this, which is that there was a predicted reward, but it didn't come. And so that's why it's called reward prediction error, because the error is assuming that there was going to be a reward and then there is none. And over time, if you continue to give the tone and no reward, the entire thing will flatline and everything will look just like this. And the neurons will stop responding to the tone at all. Okay, so this is classic dopamine. This is what everyone thinks dopamine is, and this is how dopamine has been treated since 1997. But more recently, there have been really, really in-depth studies looking at dopamine in aversive stimuli and aversive responsiveness. So we're going to look at panel G first. You can tell this is a more modern paper, but it's the same concept. In this panel, every row is a neuron, and its amount of firing is indicated by the color wheel. Basically, yellow is the most, and very dark blue is not at all, which just means baseline level firing. So again, what you see is that in the first trial, there is response to what in this case is foot shock. And then in the last trial, there is response to the tone that predicts it. So in terms of the learning, the dopamine is behaving the same, the dopamine neurons are behaving the same. But in terms of what the stimulus actually is, that's what's really exciting about this work, because the stimulus is foot shock, which is not only aversive, right? It's not only something that the animals want to avoid, it actually is probably slightly painful. And so panel H is a quantification of what's shown in panel G. And then in panel I, this is a single animal in two individual trials. And what you can see is when they don't receive a shock, after they're trained, they respond to the tone and then receive no shock. And this continues to decrease. And the reason this is important is that if these neurons were still responding to reward, you would expect the reward of not getting a foot shock to cause a spike in dopamine, but you don't see that here. So these neurons are really responding to the aversive stimuli alone, and that makes them quite special. And so, all right, so I'll show you a little bit about my data in a second. But does anyone have any questions up until then? Was that clear? Okay. All right, cool. So this is unpublished data, so don't scoop me. But all right, so let me tell you what the experiment is. My mice are running on a treadmill, so we can record their motion activity, their acceleration and deceleration. They're water restricted, so the reward in this case is they have a little water spout and they get little drops of water to drink. And then the aversion in my case is actually an air puff to the eye. So if you have glasses and you've ever been to get your eyes checked, they will puff air in your eye, and it's equivalent to that. So the idea is that this is actually not a painful stimulus at all. It's uncomfortable and nobody likes it, but it is not painful. And so the first idea in my lab was to basically replicate the findings that I just showed you in that paper. And there's a lot of complicated genetics behind what I do, but suffice it to say that my lab has special access to isolated populations of dopamine neurons so that I don't have to look at the entire VTA and hunt around for the neurons that I want. I can just study the neurons of interest. And so that's what these recordings are. All right, in the background of both sides, you'll see a black line with a gray sort of border on it. That's the motion of the animal. So moving upwards is acceleration and downwards is deceleration. And you'll notice both for the reward and for the aversive stimuli, the animals slow down on their little treadmill. However, and then the purple is the neural activity. So these are the dopamine neurons from the VTA to a specific nucleus in the nucleus accumbens. And you can see that with reward, my thing went off. The thing about testing on mice is that mice are almost completely different from a person. Okay, would you like to expand on that question? It might be inaccurate. What might be inaccurate? On drug tests. Okay, these mice are not on any drugs. I can maybe speak to that in a minute. I have a slide on morphine circuitry at the end. Okay, so I guess what I will say is that it's absolutely true that mice and people are not the same. I think that's always important to keep in the back of your head. However, at some point, we have to be able to do these studies. The best thing that I can say is that so far, everything that has been found in this particular circuit in mice can be replicated in monkeys and can be replicated to the extent that you can replicate invasive recordings like this in people. It can be more or less replicated in fMRI research. Beyond that, I don't know that there is another good control to say that what you find in mice is similar to what you find in people, except to ultimately try things in people. Yeah, okay. So I hope that addressed that. Anyway, what you can see is the neural activity in response to receiving the reward is basically unchanged, while the neural activity in response to receiving the IPUF, you see this little increase. So this is really exciting for us because we're looking at a little tiny dopamine neuron population that was thought not to exist for 25 years. So, and then just as a control, for example, one of my controls is to look at all instances where the animals speed up really rapidly or accelerate rapidly to see that nothing is happening. And the reason controls like this are important is I told you at the beginning that another classic function of dopamine is motor control and motor decision making. So we really want to make sure that other functions of dopamine like reward and motor function are not sort of clouding this finding. Okay. So here's a pharmacology slide. I know that this is really busy and ugly. Unfortunately, there is not really a better one. So this is, again, now the VTA is on the left and the nucleus accumbens is on the right. And here is where morphine is considered to act directly. You'll see that morphine does not act directly on the dopamine neurons themselves, and yet giving morphine will cause a dopamine neuron burst, as can be shown most obviously in animals and slice prep. However, they have done PET scan, which I can explain more in depth, but it allows you to look at the binding of a receptor, the amount of binding of a receptor in a person's brain, and they have done PET scan studies in terms of the binding of morphine in people. And so what morphine actually does, this red neuron here is called a GABA neuron. You can think of it as a stop neuron, and you can see that it's telling the dopamine neuron to stop. One of the major things that morphine does is it stops the stop neuron from firing. So it turns off the stop signal and then the dopamine fires more. And so more dopamine gets into the nucleus accumbens and gives us that reward signal. So it's kind of interesting or exciting in my case that the same neurons that we're looking at here that have, that have implication in chronic pain and aversion are also here in this well-established addiction circuit. And I want to draw attention here to see that ethanol, nicotine, morphine, and cocaine all have functions in this circuit right in this area. So if you read in the press, for example, a Facebook addiction has the same brain circuitry as cocaine, or chocolate does the same thing to your brain as cocaine. What they mean is when they looked, they found activation in this area, which, as you can see, is activated in with ethanol, nicotine, morphine, cocaine, MDMA, all kinds of drugs, and also all kinds of other things. Chocolate actually specifically has function that directly activates dopamine neurons. Okay, so that is sort of the end. Does anyone have questions? So is there a solution to this drug epidemic? Right, so one of the things that we're trying to do is to tease apart what opioids do that causes analgesia, that takes away pain, and what opioids do that makes them, that gives them such abuse potential. And theoretically, there should be a way to disassociate these things. And so one of the reasons that we're going after this tiny, tiny population of neurons to see if they're aversive responsive, and if we can look at that, is to see if the dopamine neurons that are implicated in pain are a little bit different, or at least can be isolated from the rest of the dopamine neurons that only have rewarding stimuli. So a dirty drug like morphine that will activate all of them without a care will cause both analgesia and be very addictive because you have that massive reward signaling. But ideally, the ultimate goal is to be able to target only the pain without targeting other addiction circuitry and other reward pathway. So that's the idea behind this science. In terms of practical public health solutions, I have plenty of thoughts on that, but I think that they are not relevant to this talk. Yeah, you can do that. So for cocaine, you can do that. You may have heard of something called a methadone clinic. That's kind of old-fashioned, but we use similar drugs today. So for cocaine, they may use the cocaine itself, although my understanding, at least in this country, is that we use a derivative that isn't so volatile, because a lot of the abuse potential of a drug can be predicted by how rapidly it gets you high and how quickly that high fades. And so, at least in the US, as far as I'm aware, we use a derivative of cocaine that changes the slope of that so that you don't suffer the physiologic effects that can cause people to have cardiac arrest if they go cold turkey, but that you aren't really getting that same peak of the high. But in terms of opioids, what we have are, like I said, methadone clinics, if you've ever heard of them, or we use naltrexone or naloxone now more commonly. And what these are is that they're partial agonists, so they kind of do what morphine does, or heroin, or fentanyl, in that they attach to the same receptors, but they don't aggravate the receptors to the same extent, and so you don't get the same level of high, but they activate them just enough to prevent those really severe cravings that people have for a drug that they have been misusing. And so that helps people who are committed to sort of overcoming their addiction that helps them because then they're not sort of having this really strong physical urge to do the drug constantly, and they have a little bit more control over that. So that's what we have in the US, and it's a great idea and it helps some people. The major issue is that you have to do it every single day and someone has to watch you take it. So it can be impractical for some people to pursue that route. But absolutely, naloxone and naltrexone are used in exactly that way to help people with opioid addiction. And like we talked about, I don't know, maybe last week or 2 weeks ago, you can also use high doses of them in people that have already overdosed on opioids. And that is because they have a stronger affinity for the opioid receptor than morphine, heroin, and about equal with fentanyl, so that they can knock those off the receptor and have that reduced effect, which is not as lethal. It doesn't suppress your breathing, for example. Okay.
Thanks, Sage.
I think we might talk about COVID now.
Do you want to slide share, Celeste, or should I do it?
First, let me unmute myself.
Yeah, that's a
do you have the slides open? I just need to pull them up.
I have them open. I'll just do it then. Let's see.
Okay. And then we can tag team from there.
All right, let me close my work. Nobody needs to see that much PHI. All right. Let me start the presentation then. All right. I guess you guys can keep an eye on the chat then, because I cannot see it. See if I can pull it anyway. So we'll talk a little bit about COVID today. Celeste and I will probably try to tag team this, and maybe Sage and Stephen will jump in if they inevitably know more about some of these topics than I do. So I know that the coronavirus can be a little confusing, and there's a lot of terms that get thrown out there. And this slide just goes over some of the basic terminology that was used. So the coronavirus is a general term, and that refers to a family of viruses, coronaviruses, and they're given this name because corona means crown, and they have these kind of crown-like projections. I guess you can sort of see them here, and it would probably appear better on a, oh, Ryan's got to go to his exam. Good luck, Ryan. And there are other types of coronaviruses other than the one that was commonly referred to as the coronavirus in the literature right now or in the news. SARS-CoV-2 is the name of the virus that has caused this pandemic. It's a little bit of a mouthful. It stands for Severe Acute Respiratory Syndrome Coronavirus 2, and it was formerly known as the 2019 novel coronavirus, or 2019 nCoV. And then COVID-19 is the disease caused by this virus. So I know it's confusing. I wouldn't worry about that too much. I'll probably interchange them all throughout this presentation. This presentation is in general about this particular SARS-CoV-2 virus and the disease that it causes, COVID-19. All right, so a bit of history. You guys are probably too young to remember it, but there was another closely related virus, SARS-CoV. That's why this one's SARS-CoV-2. And SARS-CoV, or the SARS epidemic, was a closely related virus that caused an outbreak just called SARS from in the early 2000s. And this virus originally was thought to be really dangerous, and it was really dangerous. It has a higher mortality than the current virus. But it's kind of spontaneously disappeared and then never reappeared, which is kind of surprising. And I don't think it's totally unexplained, but it did give us a little bit of a heads up on this current coronavirus, which is very closely related and has helped to jumpstart a lot of the research going on here. There's another related virus called Middle Eastern Respiratory Virus, or MERS. This is not as closely related, but this one has had really entered the news in 2012, and it has annual outbreaks. So every year, a few people get it, and the total infected in the last 8 years is around 2,500 known infections. And by far the most common coronavirus infections are just the common cold viruses. There are other causes of the common cold, but coronaviruses are one common cause. So there's a bunch of different coronaviruses. Where did they come from? The answer is they're usually from bats or civets or other small rodents. And then they're passed through a secondary carrier and then infect humans from there. So there's a few others. You can see a bunch here. There's a lot that are not infectious in humans. These blue boxed ones are ones that cause common colds. And you can see that they either come from camels or cows or who else who knows? Nobody knows. And then SARS, the original epidemic in the early 2000s, came from a small cat-looking animal called the civet, and it was also seen in domesticated cats. And again, not all these viruses are really bad in humans. For example, SARS-CoV is really dangerous in pigs, but has never been shown to infect humans. So the current thought on SARS-CoV-2 is that it comes through an unknown carrier animal, but originated likely in bats.
Okay, I guess I can go over this slide. So before we continue more and expand more on like the specific characteristics of COVID-19 coronavirus or SARS-CoV-2 itself, just take a moment to introduce what a virus is in general. And you may have already read about this in biology class or somewhere else. But basically, a virus is an extremely, extremely small biological molecule. Like, it's hard to say whether or not a virus is truly alive or not, as first, it doesn't really contain its own energy for itself to replicate, and it requires a host cell in order to propagate itself. And you can think of it as like a tiny, tiny particle that just contains genetic material that is kind of designed to hijack its host cell. And just from the scale here, you can't see my mouse because this is not my screen, but like the flu virus is already 10 times smaller than a bacteria. And that is already 10 times smaller than your red blood cell. So it's very, very small. There's not much there. The structure is fairly simple. And if we go to the next slide, it gives you some examples of like different flavors that viruses can come in. But the overall idea is that there's some sort of outer envelope that we usually call a capsid. It can encase a membrane that then encases the genetic material inside the virus. And this genetic material can come in forms of DNA or RNA. So DNA and RNA are just different types of nucleic acids that can encode for genes. And depending on the type of virus, so for example, a bacteriophage on the left, it kind of looks like a little alien with legs. And like the legs can recognize the specific bacteria cell host and help to inject the DNA, the viral DNA into the bacteria and subsequently infect the bacteria. Whereas other viruses like tobacco mosaic virus specifically infects tobacco plants, and HIV, human immunodeficiency virus, specifically infects humans. And this virus has RNA as its genetic material. So I talked a bit about like this process of infection. So if we go to the next slide, we can see how this, or an example of how this might happen. So the envelope of a typical virus is designed to first kind of protect the genetic material inside the virus to the best of its simplistic ability, and also contains some sort of factor that can help recognize its target host extremely specifically. So in this example, a flu virus has like these little spike proteins on the outside of its envelope, and it can recognize a specific receptor on the outside of a cell. And once this kind of binding event happens, it attaches to its target epithelial cell, and the cell then engulfs the virus. So now the virus has accomplished entry into the cell. Once this happens, it can release its viral RNA, and then this viral RNA can be transcribed by, well, depending on the type of RNA, it can either be copied first and then translated into proteins that form new virus particles, or it can be directly transcribed into virus components. So once this happens, the virus takes over the cell, and the cell is now just producing more viruses. Eventually, the viruses overwhelm, the new virus particles overwhelm the cell, and they just escape. And usually, depending on the type of virus, this can either kill the cell or like slowly release viruses and just impair the cell. But the end result is that you get uncontrolled production of more viruses that can then swim around in your body and infect more cells. Okay, so now we can move on to the specific structure of SARS-CoV-2 as far as we know. So like the other coronaviruses, it has this like spherical shape and these crown-like projections outside of the envelope. These are termed spike glycoproteins that are attached to the virus membrane. So the membrane is actually a bilipid layer, and so lipids have this property known as being hydrophobic, in that they are not well dissolved in water. So knowing that lipid bilayers are not well dissolved in water, how might hand washing with soap affect the lipid membrane? Like, what do you guys think soap can do to a virus's membrane, outer membrane? Do you have any ideas? Maybe that question was worded vaguely. Okay, doesn't soap, the rubbing motion of hand washing, help dissolve the lip capsule and render the virus useless? Yes. So you got a major component correct, in that soap definitely helps to dissolve or kind of destabilize the membrane. And remember, I mentioned before that the purpose of the membrane is to protect its genetic material so that it can enter into the cell. So dissolving it basically does render the virus useless. And the rubbing motion definitely helps soap to reach every part of your hand. But the property of soap is that it creates these amphiphilic, it's like an amphiphilic material. So it can kind of solubilize lipids and make them soluble in water. And that kind of destroys the integrity of the lipid membrane. So actually, washing your hands with soap and water is likely more effective. Well, it's the same principle is used in hand sanitizers as well. But yeah, so soap is highly effective at washing away virus. So don't just use water. And what do we have on the next slide? Okay, so where did it come from?
It's a little bit.
Mike, do you want to mention this one?
So I think you guys are all familiar that it's likely from Hubei province in China. And this is also saying that it's very likely that it came from bats. Just so you know where Hubei province is, that's where it is in China. A little bit inland and right in the middle. But mostly the slide was here, where did it not come from? There's a lot of things in the news right now, but maybe it was engineered in a lab and intentionally released or unintentionally released. While it is possible that there was a lab specimen that got out, there is very strong evidence that it was not intentionally engineered in a lab and was likely arose naturally in animals. And a lot of this evidence, you can read about it if you'd like, but a lot of it is relying on observations that all the essential features of the new virus are found in viruses in animals in nature. So coronaviruses that are related in other animals that have not infected humans yet. All right, so we can talk a little bit about the infection progression. And again, I think this is fairly well established, but there's still potential that it's not 100% right. This is all relatively new. You can see this is published in the middle of April. And we see someone that is exposed to coronavirus and also starts to develop the disease. The first couple of days, they'll have an asymptomatic nasal infection. And again, this is the way it progresses in a majority of patients. There are patients who progress slower or faster depending on their own health and other factors. So over the next couple of days, so maybe 3 to 5 days after the initial infection, there's a symptomatic upper and conduction airways infection. So around 80% of patients, we think, and this depends on the country and other factors, but a lot of patients just have a kind of a upper airway infection, some coughing and maybe some discomfort, and then they get better. So that's good that most people don't get severe infections. But these patients also are carriers of this virus and may not think that they have the coronavirus, so are capable of spreading it. And then in a smaller percentage of patients, they progress to a severe lung disease. So this is a alveolar disease, and we talked about the alveoli last time. We also talked about the preference. We also talked about type 1 and type 2 pneumocytes here. And I'll remind you that the type 2 pneumocytes are the ones that generate the surfactant that keep the lungs pliable. So the SARS-CoV-2 virus strongly prefers type 2 cells, is what we believe. So symptoms, I think you guys have probably heard about this in the news already, but fever is a prominent symptom, as well as sore throat and cough, as you'd expect of an upper respiratory or lung disease. As it progresses into the lungs, patients start to experience shortness of breath. And then there are some other more interesting symptoms, which are a little bit as less common with other. So muscle aches, they can be seen in flu as well, but are seen in COVID-19. An uncommon symptom, which is seen in many patients with coronavirus, is a loss of taste or smell that's unexplained, and particularly the loss of smell is pretty strongly associated with this disease. But patients can also experience symptoms elsewhere. Diarrhea and headache are some of the more common other symptoms.
And purple swollen toes, right?
Yeah, purple swollen toes, and actually in children, I probably made this slide a couple weeks ago before there were any purple swollen toes or before it was known. But yeah, more recently there's been some purple swollen toes, I think especially in younger people. And then in the last week or so, there's been evidence that there's an inflammatory-like disease, especially in young patients and especially in patients who are formerly healthy. So people your age and even younger. And it was kind of thought to look like another disease called Kawasaki disease, but it has some slightly different presentation. So yeah, it can, and this is kind of contrary to what was formerly thought to be true, which was that children were largely spared the coronavirus, or at least the symptoms of the coronavirus. And so I think that's still largely true. Kids tend to have milder disease, but definitely in a small fraction of kids that get this disease, they have extremely severe symptoms and even die. We've heard a lot about 2-week isolation. So if you're exposed, or if you have the disease, that you should spend 2 to 14 days. Sorry, you should spend 14 days or 2 weeks in isolation. And this is relatively simple. It's because that the symptoms can appear anytime from 2 to 14 days in the majority of cases, and that people remain contagious for a while after they develop symptoms. The challenge with this virus versus others is that people are actually carriers and shed the virus, so they can spread the disease without actually having had any symptoms, or while having had extremely mild symptoms that they wouldn't associate with the virus. So another thing that you've seen probably in the news, or at least heard about, maybe not, but the people saying that the coronavirus is going to go away as soon as it gets hot. So there is good reason to think this naively, and you could say other coronaviruses exhibit this type of seasonality, and you can see here, this is a fairly local plot, so I think it's from like Germany or Switzerland or something like that, but it shows that there's a much higher positive test rate when they're testing for coronavirus, and this is the common cold causing coronavirus, not the current epidemic. But you see that these other types of coronavirus that cause the common cold do have a pronounced seasonality, so they're much worse in the colder weather. And this could be because of intrinsic factors about the virus, or because people spend much more time close to each other indoors during these winter months. But in the case of this virus, there's a lot of evidence that it will not go away when it gets hot, unfortunately. And there's some things that you guys could easily come up with here. Why won't it go away when it gets hot? Well, look at places that it's not where it's hot right now. Australia, California, India, all of these places have really high and growing numbers of infections. And throughout the Southern Hemisphere, it's summer right now, the coronavirus is spreading widely throughout the Southern Hemisphere. So my personal view is that it will not just spontaneously go away when it gets hot. And this can be contradicted by other powerful people, but I think the future will bear out my prediction. Yeah, coronavirus is not an extremely complicated disease to take care of people with. That doesn't mean it's easy, but the actual treatment is relatively simple at this time. Primary treatment is to make sure that the patients can have adequate oxygen. And this can involve supplementing them with oxygen, and then when they become unable to ventilate for themselves, to put them on a ventilator. And you've probably heard a lot about ventilators lately, but essentially they help the patients breathe by breathing for them. So this is not some place that people want to be. Essentially, you'd have to be sedated, and then they put a tube down your throat, and they do your breathing for you. But it can allow people to bridge over while their immune system helps defeat the virus, and then they can be released afterwards, where otherwise they wouldn't have been able to breathe and would have died. There are some other treatments which have been in the media more. A couple of these are also in trials in the United States or other places, and Northwestern is actually doing trials of some of these drugs as well, actually all of them. So one of them is remdesivir. This is a drug that inhibits that enzyme that Celeste briefly mentioned, we know that the coronavirus is a positive single-stranded RNA virus. So it requires an enzyme that it carries to make RNA from RNA so that it can be translated. And this is a feature that's in common with Ebola. And so this drug was actually designed for Ebola, but shows promise in coronavirus infections. Because if you can, humans don't have a need for this virus, so it's an easy virus. It's an easy enzyme to target. Not easy. It's a rational or logical enzyme to target if you're trying to control infection. So that drug is in trials now. Sarilumab, when I made this slide of slides initially, it's an anti-IL-6 receptor antibody. I wouldn't worry too much about what IL-6 does. But it's thought to reduce lung inflammation. And there was a trial that was a fairly large trial in the United States where sarilumab was not shown to be helpful in patients who just needed oxygen. So these are patients who have mild illness. So that trial was actually, I believe, discontinued. But there is a trial continuing in ICU and ventilated patients, so in severe patients, to see if this can help. And then lastly, the drug that's probably gotten the most press because it's been pointed to by the president repeatedly as a panacea is hydroxychloroquine. This is a malaria drug. Right now, there's really no strong scientific evidence that I've seen that it helps very much. There is strong scientific evidence that that it can cause dangerous heart rhythms. So actually, most of the trials using hydroxychloroquine have been curtailed or shifted towards very ill patients. But it is talked about a lot.
Yeah,
You have a question about the nanoantibodies from llamas or microantibodies or whatever.
I can't speak broadly on this, but I have seen reports of this. I'm not an expert in the antibodies from llamas. Does anyone else know more than me?
Celeste.
So this is pretty, as far as I know, it's the very recent development. So I can only offer some conjecture based on my scientific opinion. First, I just want to reiterate that, like, especially in this day and age, news of any sort gets out very fast. So this is the most important time for everyone to read critically. So if you hear a report about someone using something, it doesn't necessarily mean that that's the right thing. For example, hydroxychloroquine. As far as the llama, yeah, exactly. So I think this is a prime example of being a critical reader and how that might benefit you in the long run. As far as the llama antibodies go, with all new developments, they're extremely exciting. But at the same time, we need to remember, like, a lot of these initial observations are observations, and they require a lot of study and further development and follow-up to really substantiate the findings. I'm not really familiar with how these antibodies might work or how they might be purified, but to raise an example of another treatment method that's been discussed, which is plasma transfer from humans with antibodies against coronavirus having overcome an infection themselves, and testing that kind of treatment in patients with active infection. So this is a methodology that's been tested before with varying results because antibodies are highly, highly personalized, not just to species, but to each individual. So it's very hard to predict how they might pan out when you put them in another human. So I would say for this story, like, it's certainly extremely interesting, and perhaps you can isolate a component of the antibody that can then be developed into a drug that won't be necessarily targeted by an individual's immune system. But yeah, so I'm not sure if that answered your question, but just there's definitely more to be researched. And then the second question is, isn't hydroxychloroquine prescription only?
Yes.
I believe so, because it's an anti-malaria.
Yeah, all of these drugs are prescription only.
Nick?
And
You could probably
hydroxy,
get oxygen, but even getting pure oxygen is not super easy, and you're not going to be put on a ventilator unless you're under medical care. Sorry, Sage. How does Trump get them? Being the president gets you access to a lot of things. Also, being a billionaire gets you access to many things. So you've seen a lot of examples of NBA players being tested and all sorts of stuff. There's unfairness in the world.
I will say that what we're talking about the dangerous heart rhythms and hydroxychloroquine is why that even as an anti-malarial, it's not used in this country. So, because there are other treatments for malaria.
But I think the other...
If a doctor prescribed it, could they risk a malpractice suit? So if they're prescribing it to Trump, yes, because he has no malaria and no COVID. The way that doctors are prescribing it to other patients, as far as I'm aware, you guys can correct me, is under what's called compassion care, which means that this is an unprecedented disease and we don't have good treatments for it. So a patient that's really, really sick that you're afraid is dying, you're allowed to petition to give them treatments that you think may have efficacy in order to try and prolong their life and save their life. And that is the way that hydroxychloroquine has been prescribed for COVID patients in the United States up until this point. In other countries, they have
prescribe it as well.
Yeah, in other countries, they have different protocols, but that's the major mechanism by which it's been prescribed in this country. I pulled up the original cell paper for this llama stuff if we want to look at it. It's the same idea as what Celeste said. It's just that their antibodies are really, really small.
Yeah.
So they're less likely to cause some kind of like secondary allergy or something, I guess.
They're a little easier to make in other constructs, like I think like bacteria. They're so small.
Can you speak up, Steven?
Oh, sorry. I think it's fine.
Yeah.
You can they're a little easier to make in like bacteria because they're so small, and a lot of times their structure is not as complicated.
Yeah, so
not that they'll work, but you don't know if they work. But.
I'm not going to be first in line for llama antibodies, but maybe I'll be fifth. Yeah, so there's other ways to get hydroxychloroquine. One is to enroll on a study. So there are a number of very, very large studies that are enrolling across the country. So patients can ask to be enrolled in studies, and doctors have wide latitude to enroll patients in studies. That said, since hydroxychloroquine has been shown to cause heart rhythms and have very little efficacy, at least so far, there's certainly ethical questions about whether you should be enrolling patients if it's not actually helping them. The other thing that doctors have wide latitude to do, and this is less well known, but to prescribe drugs off label. So a drug that's prescribed, such as hydroxychloroquine, has some use and has also been shown to be reasonably safe. So it's not going to immediately kill you if you ingest it for most drugs, some of the chemo drugs notwithstanding. So doctors are allowed to prescribe drugs, maybe for reasons that are not the intended and approved reason, but if they have reason to believe that they could help in some patients. And actually a lot of drugs are prescribed this way routinely. One example is some contrast agents for imaging are actually approved as other drugs. There's a new one called ferumoxytol, which is basically iron. It's approved as an iron supplement, but doctors are able to prescribe it for imaging studies because it makes really nice images and doesn't harm the patients. So it's not FDA approved for this use, but it's often used. So doctors have pretty wide latitude. I think the legal implications of this, I'm not sure of exactly. I think people can sue you for malpractice for a lot of things, but I think trying to operate, as often is the case, people do operate to cover their butts. But I think it's kind of sometimes people are doing things kind of at the moment, trying to do the best things for their patient.
Also with the suit and COVID in particular, some states have put in practices that you can't sue like doctors and nurses caring for COVID patients because the conditions are so dramatic. And They're often caring for too many people. I think that was
Yeah, exactly. That was another thing I was going to mention. A lot of these projects a lot of drugs are actually used in very high concentrations in poisons and other uses. So I think this is true. There was at least one guy that took hydroxychloroquine. I don't know if it was actually hydroxychloroquine that he was taking or he misread the label of what the fish cleaning was, but
yeah.
I don't think it was actually hydroxychloroquine or it had other ingredients mixed in. I don't know exactly that story. But for instance, a lot of like rat traps have blood thinners in them. So there's a lot of these drugs are used for a lot of different things. Yeah. Well, I'll move on then, unless there's more questions.
Yeah, they were taking chloroquine phosphate. And I think another thing to note from that example is that when you dose hydroxychloroquine, you do it very carefully. So even if it had been hydroxychloroquine, the fact that they weren't under, they didn't have an expert looking at the dosing and watching them for signs of disease, I think is the second issue with that. But yeah, don't take drugs that are made for animals, especially not fish.
Yeah, you're not really like a fish in too many ways other than you're alive.
Or like a fish tank.
Yeah, it's not even made for fish. Yeah, I would be very cautious about putting anything that's not food into your body that's not prescribed by a doctor.
And even some food.
True. Anything that doesn't go bad. All right. After that aside, I added a slide recently just asking about social distancing and masks. Why do we think social distancing is important? Why did we choose the numbers? Not we, I'm saying we in general, I'm not involved in these decisions, but why 6 feet? And do masks help? So I think the disease appears to be spread through respiratory droplets. It's really hard to test this in humans because you have to infect people to do it. And it's hard to do it under controlled conditions. There aren't very many people volunteering to be infected with COVID, nor would you be allowed to conduct that study. But essentially, in other similar diseases, these droplets and other simulations and computer models, these droplets have been shown to travel 3 to 6 feet before settling to the floor. But obviously, this is a lot of assumptions. There's like wind can change things, temperature, all sorts of stuff. So this is just a general guideline. In general, if you can stay away from other people, that will keep you more likely to stay healthy. And we know that the coronavirus spreads most easily, and isn't true in general for coronaviruses, most easily when people are in close proximity. So if you're stuck inside with somebody, you're much more likely to contract the disease. And I think that's pretty intuitive. The question, do masks help, is a pretty charged question right now. The CDC position, so that's the Center for Disease Control, though they've moved around on this a little bit, and I think they've received some justified flack for that. Their position is that you should wear a mask, and whether that's cloth or just a normal kind of over-the-ears surgical mask, to prevent transmission in the case that you're an asymptomatic carrier. Right now, the CDC is saying nothing about whether or not it will actually protect you from getting the coronavirus. It's conspicuously absent from their statement. I'm probably not qualified to offer anything here, but I will tell you that I wear a mask when I go outside, and would recommend that you do. But again, the Center for Disease Control has not recommended anything there. I believe that the Illinois governor and Illinois state has issued a mask wearing order. So you must wear a mask if you aren't able to abide by social distancing guidelines. So yeah, you should be wearing
And if you're inside. In Chicago, if you're in a store or restaurants aren't open. So basically, if you're in a store, you have to be wearing a mask and attempt to be 6 feet away from other people, but you must be wearing a mask. And most stores will require you to anyway.
Yep. Yep. And then another just a brief aside, you've seen a lot about N95 masks. The CDC's position still is here that these need to be reserved for healthcare providers. These definitely help with spreading of the virus because they filter out 95% of very small particles. So certainly those do help. And yeah, I guess I'm not allowed to conjecture here because I can't provide you medical advice. You want to talk about COVID testing,
Yeah.
Celeste?
Okay, so there's a couple of different genres of testing for COVID. One is to test for active disease, and one is to test for resolved disease. So the PCR test, have you guys heard of polymerase chain reaction? Does anyone want an explanation? Okay, sounds like everyone knows what PCR.
Sounds like an emphatic yes or no.
Okay, sounds like everyone knows what PCR is, so that's great. So basically, let's just test for the presence of COVID-19 mRNA. I guess SARS-CoV-2 mRNA to be technically correct. This is a fairly simple test. We could theoretically do it in the lab. And one thing to note about it is that it's extremely sensitive. So even a few copies of this mRNA can be detected by PCR, which can actually bring up some difficulties because there have been reports of patients testing positive for SARS-CoV-2 mRNA even after possible resolution of their symptoms. So that's still a bit of an open question, but some say that like this could just be the final residual particles remaining in the system waiting to be cleared out rather than like a second infection. But anyway, I digress. So there's the PCR test, and there's various other tests for coronavirus that have been developed, but the one that's been used most commonly in the US is the PCR test. And another important test is the serology test, where they test for antibodies directed against COVID-19. So basically they have a surface with some COVID-19 antigen or like parts of the virus, and then they take a bit of your blood and see if it reacts against the virus. So I added this little diagram to show you how antibodies might come about. So when you first get infected with any virus, your cells kind of produce a panoply of antibodies, and eventually you select for the one that is the most effective against the actual virus. And then the cell that produces this super effective antibody is then preserved as a memory cell, so that when you're exposed to the infection a second time, you don't actually feel the symptoms because the cell's like, oh, I've seen you before, and they make the antibodies against the virus. So the test is kind of like mimicking this second time infection on the bottom. And this test for serology is actually pretty interesting to conduct nationwide if possible, and I believe the NIH has already started this study because it can show you like how many cases, or at least provide an estimate of how many cases of coronavirus are actually out there as opposed to the ones that kind of made it to the hospital.
Cool. There's a few things that we don't know, a lot of things we don't know about the coronavirus. I'll just summarize a couple of them here, and then we can take questions from you guys. We're not the world experts on coronavirus. We know a little bit about it. But what we don't know how the disease got into humans. So evidence points to a bat coronavirus as the most immediately related, but we're unsure of the intermediate host. So what it went from a bat to another animal and then to us, most likely. We don't know what that other animal was, at least not that I've seen. So how come some people get very sick and others have no symptoms or mild symptoms? Again, this is something that's being heavily researched right now, and there's been a lot of evidence coming out in the last few weeks, even the last couple of days, about who gets sicker and who doesn't get sicker. The evidence points to that people with existing health conditions and people who are elderly having worse outcomes. You've probably heard a lot about elderly people getting sicker. So people who are obese tend to need a ventilator more often and have worse outcomes. And men also tend to have worse outcomes. So tends to be the better health you're in, the less likely you are to have really severe disease and need a ventilator and die. I think that makes some sense. You have some physiologic reserve of like how much of an insult you can take to your system before you need help. But again, this is still largely unknown on who is resistant and who is not resistant to the coronavirus. In the chat. ACE2, yeah. ACE2 is the protein that's targeted by the coronavirus for uptake. And it's widely expressed in the body. One prominent place is in the lungs, but it's also expressed in the heart, in blood vessels, and other places. And so there's a lot of interest right now in trying to see what other outcomes are from the coronavirus. Obviously, the primary concern when you have an acute disease, so a really severe disease, it's a sudden onset, is making sure the patient survives as best they can. But there's a lot of worry about long-term heart effects because ACE2 is prominently expressed in the heart, also kidneys, liver, blood vessels. It's expressed everywhere. So we don't know as much about the virus's effects on these other organs. It may not have any effects, but it may also. So I think there's going to be a lot more studies going on in the future, trying to see what other effects the coronavirus has other than lungs. We certainly know there are some cardiac effects, but these are less well elucidated than the lung findings. That's a good question, though. Other things we don't know, how long until there's a vaccine. I've seen a lot of promises that seem startlingly short to me. As far as I know, the world record for fastest vaccine development is something like 5 years. So getting one in 8 months would be a world record many times over, but we'll see. I think
some of those vaccines are just the vaccines that were already in preparation for SARS, for the original SARS.
Correct. So we have a bit of we have a bit of a head start here, and there are a huge variety of different types of vaccines and different approaches. So we'll see. Hopefully there's some success there, and hopefully relatively soon, but we don't know when that will be or if that will be, actually. We were asked how long until things go back to normal. Nobody knows the answer, and things may also never go back to the way they were. They may be slightly different, so we'll see. I probably put this out of order. Heart problems in the news. Again, the virus uses the ACE2 enzyme to enter cells, so it's expressed all over the place, and there's a lot of interest in long-term effects in these organs. And that's the last slide of our presentation. So if you guys have questions, other things, we're happy to take those. Hopefully that was helpful or at least informative. Wow, it's already 5:20 too.
Yeah, we
Rambling on as
forever.
usual.
Yeah.
Sorry.
No, that was me.
Chatty.
That was a team effort. That was a team
Team
effort.
rambling effort.
Yep.
If you guys don't have other questions, I think we can talk about other things that we'd like to talk about in the future or things that you'd be interested in hearing about in the future. My observation, as well as that of some of the other mentors, is that you guys have considerable interest in medical school and what it takes to get into medical school. We have considerable experience looking at medical school applications. We've also all successfully applied for medical school. So if you guys are interested, we could give a presentation on kind of what the basic requirements are for medical school, and kind of what our recommendations are about going about fulfilling these requirements. Obviously, there are a million different ways to go about this, but there are some general things that we think could be important. Okay, so at least one person, Melissa, thinks that would be helpful, so we can do that. Yeah, so we're not the be-all and end-all on med school admissions, but certainly I think we can provide a little insight there. What else did we talk? Oh, we talked also about medical professions. We did a short, or not that short, talk about different not medical specialties, about healthcare professions. So hopefully you guys thought that was relatively helpful. I know we have a lot of interest in different professions in this group.
I think we were also maybe thinking about different specialties once you are a doctor.
Yeah, correct. So if you guys are interested in hearing about different specialties that doctors go into once they have finished medical school, we can talk about that as well. If not, tell us and we won't talk about it.
The fun thing is, since none of you talk, whoever answers gets the say.
So it looks like Anna has spoken for the group there as well. Anything else that you guys are interested in? I mean, we're happy to talk about pretty much anything. Celeste and I have not talked about our research yet, so we could also talk about that. If you guys are interested in that, we have very different research.
Yes. Do you guys want to hear about imaging, or do you guys want to hear about biochemistry?
There's only one right answer.
There's only two options.
Or neither one.
Or none. That's also an option. All right.
Looks like
Yeah.
neither one.
Yeah.
There's no following the dawn of multicellularity, so.
Oh, biochemistry.
Okay.
Biochemistry.
You're up next,
wow.
Celeste.
Celeste!
I guess I either won't talk about my research or I'll go last.
No, you can still talk about it. I'm curious.
You're curious?
Yeah, what is it that you
Yeah,
do?
me too.
I know you're the tech guy.
I do IT work mostly, unfortunately. No, I can talk about my stuff last. My stuff is probably the least related to everyone else's. I do a lot more computational stuff and some more stuff in AI and also in imaging. So
Low-key, I would recommend in general to focus on computer-based skills as you proceed in your life. As someone who,
No
as you
one
can
ever
see, I do
said-
a lot of wet lab stuff.
nobody
Like-
ever said, that guy is too good with computers. Sage is in lab. She is an essential worker,
I am.
unfortunately.
And I was moving my brains from one bath to another just now.
So yeah, Sage is casually working.
Oh,
Yes, I was.
Cool. If you guys don't have any other questions, I think that's probably it for today. We'll try to talk. Maybe Celeste will talk next time if she has the time to prepare. Otherwise, I can talk. And
Yeah, I
we
can
can
do
talk.
next week. And
Okay,
then we
Cool.
can
You'll hear
maybe
from Celeste.
talk, because I'm definitely not going to take an hour and a half to talk about my work. No one wants to see
I
that.
don't want to hear about it for that long.
Definitely not. That's way too in
20
depth.
minutes tops.
I mean, I can definitely go on for an hour and a half, but you know.
Do
So,
we know how many weeks
How
we
many
have
weeks
left?
left we have? I don't think that's been decided. Maybe Blanca
Yeah.
can speak on that. I think
We are
mostly-
still undecided on how many weeks we have left, but it will be something we discuss going further.
Sure.
Do you
I
have an
think
opinion?
I think our view is that we're around. If we're around and you guys are interested, I'm at least happy to meet with you guys. I'm happy to talk about almost anything that you guys want to talk about. If you guys think that's helpful and entertaining and or interesting, as long as the Boys and Girls Club is willing to facilitate it. I think at some point in the next month, some of us will probably have to go back to much more regular work hours. It's already happening.
Some of us already do that.
As you can see, that Sage is still working. So I think we're all working remotely, and some of us are starting to work in labs some more. So we may have to adjust the hours if it doesn't work for us based on our actual work hours. But as far as I know, all four of us are relatively happy to talk with you guys as long as you guys are interested in hearing from us. We're happy to talk to you guys. We know you guys are probably stuck mostly at home, and if we can provide any entertainment or learning, we're happy to do that.
Cool. Okay.
So the answer is
So
no, we don't know.
yeah, I think that ultimately depends on what the next stages are with regards to schools and what reopening looks like. So unfortunately, I can't really give you a good timeline with respect to that.
And we'd love to
Um,
restart in-person stuff as well as soon as we're able, and we think it's safe. But obviously, our priority is keeping you guys safe and healthy, and so we're not going to do that until both Northwestern and the Boys and Girls Club clear us to do that, obviously.
Yeah, so we're kind of running on the fly as far as the timeline goes, but we're happy to work with you guys.
Yeah.
And
If you guys have suggestions about stuff that you want to do or want to hear about, or things we could do better or stop doing, please tell us. We're still learning. Or tell Blanca so she can tell us anonymously.
And like we can also maybe per each week, like kind of announce the topic in the email, unless that's already been done and I just missed that.
Definitely not this week because we definitely didn't tell Blanca the topic before.
Okay. Okay, cool.
So she would
Nice.
not have had the requisite resources to do.
Okay. So then maybe that might help to structure things, I guess, or just give people an idea of what they're in for. Yeah, all right. So next week I'll chat a bit about what I'm doing, and we can talk a bit more about like the different specialties within medicine.
Anything from you, Blanca?
Let's see. No, I think that's actually it for now. Thank you guys for coming to another class. I hope all the rest of your exams go really well.
Yeah, good luck with the AP.
Yeah, good
Still
luck.
some left.
But that's it from us this week. Thank you guys.